Everybody loves
Howdy, Eggs.
you do.
Hope you guys are doing well, staying safe.
To the way you
Get outside in the sun when you can, but watch those humans. Hope you have a dog or cat you can hang around with outside, or just get in that backyard, relax. So we're back. We're back with more biomechanics. Today's edition is all about skeletal muscle mechanics. So that's the focus of today. We had kind of an introduction last time talking about how muscles are constructed and how we form what we call hoop strength. And that's the way the muscle fibers are set together with the connective tissue. So we have layers of connective tissue that surround each muscle fiber, called the endomysium. Paramecium surrounds group of muscle fibers, and then the epimysium surrounds the entire muscle. So that's how muscles are packaged. It allows them to produce hundreds and even thousands of pounds of force. And when that force is transferred, it has to be done with connective tissue and with the muscle fibers working in unison. They have to work together. Otherwise, there's no way to keep the muscle from completely tearing itself apart every time it contracts. So we're going to revisit the slide from last time. It's from a series of studies that was done at the University of Michigan. And what's really exciting about these studies is they demonstrated how important proteins are involved in the costomeres. And that means the dystrophin glycoprotein complex, the focal adhesion complex, the caveolae that we talked about. These are hotspots where we have lots of cholesterol, believe it or not. So cholesterol is actually good for us as long as it's in the cell membrane where it's supposed to be and guiding the work of some of these proteins. Normally, these costomeres are able to nicely transfer forces from the Z disc to the extracellular matrix, to the connective tissue. So that way, when a muscle shortens, the connective tissue and the fibers shorten at the same time, and then the connective tissue and the muscle fibers lengthen at the same time. So we call this a parallel, and series elastic element. The series elastic element or series transfer forces goes down the muscle fibers from, this is a sarcomere, here's another sarcomere, there's another sarcomere. They're in series. You have tens of thousands of them across a long muscle fiber. So some of it is like links in a chain, in series. About 50% though is in parallel. But to get that force to the outside, because the connective tissue and skeletal muscles can't generate force. That force has to come from somewhere. So it comes from the contractile apparatus, and some of it, about half of it, get passed through the Z disc. There are proteins like desmin, etc., that transfer these forces to the outside through the costomeres. So the costomeres normally function really well. But in certain cases, like when people get older, aging, muscular dystrophies, space flight seems to affect this, as well as metabolic diseases. We see disruption in these proteins, and when that happens, then our ability to be able to transfer those forces is impeded. So instead of this being a 50-50 proposition, we might be looking at 75% of the force has to be transmitted in series and only about 25% in parallel. So what does that mean? Well, when a muscle is shortening, and especially when it's lengthening, you're going to get shear forces between the muscle fiber and the connective tissue being produced. And we need actually a little bit of shear force in order to maintain muscle size and even produce hypertrophy. But if it gets too high, then we can get delayed onset muscle soreness, chronic damage that happens with muscular dystrophies. You can also see impeding protein synthesis, for example, and we might have very poor repair. Blood flow is affected adversely. So is activation of satellite cells, etc. So again, normally it's 50-50. And in this series of studies, they looked at mice that had muscular dystrophy, and they weren't able to transfer forces sufficiently in parallel. And so that's one of the reasons why the muscles got damaged. It was basically repeated shear stress. And that was accelerated or amplified, if you will, by calcium and by oxidative stress. Oxidative stress produced by NADPH oxidase 2, otherwise known as NOx2, seems to be the trigger. So it's a cascade of events. There's mechanical damage, and then there's chemical damage. So mechanical damage is first, but that triggers even a larger chemical mediated damage, which makes it a lot worse. And because when there's a disruption in these proteins, nitric oxide synthase is not where it's supposed to be. It can't regulate satellite cell activation, can't regulate blood flow, et cetera, pretty well. So one of the things I want us to take notice again is where the myonuclei are located. They're located at the cell membrane. As a matter of fact, the cell membrane actually surrounds each myonuclei. Less than 5% of the nuclei in a muscle or related to a muscle are actually satellite cells, about 3 or 4% usually. So here's a satellite cell. It's sandwiched between the extracellular matrix, basically the endomysium, which is made of basal lamina and fibrillar cartilage, or collagen, excuse me, collagen. The satellite cell is kind of sandwiched between there, where it can become activated and become a myonuclei. It can also divide and become multiple satellite cells that can repair a muscle fiber, can even replace an entire muscle fiber as well. Okay, so I think we've looked at this before, but this is the very end of a muscle fiber. And again, this is a reminder that there are layers of connective tissue that surround the muscle fibers, the endomysium, and endotenon, which are continuous. They're part of basically the same wrapping. So if you want to think about it like maybe wrapping a cigar, or maybe wrapping around, I don't know why Twinkies come to mind. I don't eat Twinkies. But wrapping around a Twinkies or a package of donuts, it works sort of like that. That this packaging is continuous, and that enhances the integrity of the muscle. So that way the muscle fibers and the collagen fibers from the tendon work together in unison. So here's another look at this dystrophin glycoprotein complex and the focal adhesion complex. So just as a reminder, integrins and dystroglycan attach the inside and the outside of the muscle cell. In this complex, integrins and focal adhesion kinase, they can act as stress strain sensors. Calmodulin and nNOS can do that as well. I'm going to have to, they migrate. I'm going to move them over here. There we go. There's where they're supposed to be. I'm kind of tweaking things, and as I do that, sometimes things move around. There we go. So both calmodulin and nitric oxide synthase can regulate muscle hypertrophy. Interestingly enough, nNOS can also regulate atrophy. When that happens, it moves. Yep, there's Trixie again. So there's usually a guest that comes in when I'm doing this. Trixie's checking out what's out there. Don't bark too much yet. So nNOS, when there's unloading, when there's during space flight, or maybe this can happen also when someone is casted, or sedentary lifestyle, or aging, nNOS will lose its place from this complex. And as it drifts away, it goes from being an anabolic agent to a catabolic agent. It will actually turn on some proteins called ubiquitin ligases. And those are proteins that turn on proteolysis. They're going to start to chew up actin and myosin and cause the muscle to atrophy really pretty fast. And this happens, you know, within just 2 or 3, 4 weeks with casting in an ICU. So we're thinking about people with the COVID virus. One of the issues would be if someone's hospitalized, and especially in an ICU, they're going to lose a tremendous amount of muscle mass, limb muscles, as well as the diaphragm, which is your major inspiratory muscle for breathing. And when people have trouble when they're on a ventilator, it's often because the diaphragm muscle doesn't work very well. There's a lot of research related to this. The problem appears to be oxidative stress. They're trying some drugs. There's an inhibitor of IL-6, that's interleukin 6, that they're trying out at Yale and a few other places. So a number of medicines are in clinical trials, and hopefully we'll find some solutions, eventually vaccines. If we can stretch this out long enough, hopefully that's the case. But the point is that when someone's in a hospital, not only are they dealing with the virus, but you're also dealing with a loss of muscle mass, which is going to happen pretty rapidly. And it's your body's using the muscles actually as a source of amino acids, which it needs to build proteins that are going to help to fight disease, help to repair a damaged tendon, ligament, cartilage, bone, etc. So your body thinks there's trouble going on, and it's right. So it's trying to do what it can to defend the body, especially based on what our genes were like many, many years ago. So this is another look at the same complex. This is mostly the dystrophin glycoprotein complex in here. And again, just a reminder that these proteins can bind the collagen, laminin, so it's a way we secure the muscle cell to the connective tissue. And here's dystrophin. Again, this is the long protein that where there's mutation can cause Becker or Duchenne muscular dystrophy, and they're the worst types of muscular dystrophy. Dystrophin is attached directly and indirectly to calmodulin and nNOS, and that's why when dystrophin is mutated, you lose calmodulin, you lose nNOS from the dystrophin glycoprotein complex. So especially nitric oxide synthase becomes more catabolic than anabolic. So it's a big problem for people with muscular dystrophy. So this is some data also from muscular dystrophy. This is from a guy named Justin Percival, who we tried to hire a couple of years ago. Didn't quite work out. He's up at Penn now. He was at Miami, or at the University of Miami in Florida. He showed, which is really interesting, that you need these scaffolding proteins like dystrophin and dystroglycans, et cetera, to be able to construct mitochondria. So mitochondria, they're the powerhouses, if you will, for fuel and generating ATP that use oxygen in the body. They were captured organelles from, a billion or 2 years ago. But mitochondria, of course, use oxygen. They generate lots of ATP. You need lots of mitochondria in muscle, especially to reduce fatigue. In skeletal muscle, there are two populations. There's one that lives by the cell membrane. In skeletal muscles, we call that a sarcolemma. So that's a subsarcolemmal population. They live right here. Here's healthy these pictures were taken with the electron microscope, really powerful microscope that can get down just about to the nano level in terms of size, so nanometers. When we have healthy mitochondria, we've got lots of them. This is where when someone is training and doing endurance exercise, so they're aerobically training, this is the population. These mitochondria, they're the ones that actually see the big increase in skeletal muscle. There's another population that is intermyofibrillar. Intermyofibrillar. I should probably edit that out, right? The IFM mitochondria, they live by the contractile apparatus. They don't respond as much to exercise. They're there, but what we think is the subsarcolemmal population, that's the one that provides ATP for ion channels, et cetera. And there are lots of those by the cell membrane, especially when a muscle's contracting, there are ions flowing everywhere. You got to move calcium and sodium, potassium, et cetera, around, calcium, of course. So these respond to exercise training. They're also susceptible to type 2 diabetes, metabolic disease. They're also susceptible to muscular dystrophies. In the case of muscular dystrophies, Justin Percival showed it's directly related to the loss of dystrophin and scaffolding proteins. So In fact, these electron micrographs were actually taken here at Texas A&M. So this is local data, if you will. So with Duchenne muscular dystrophy, these I think are mice, or they may be larger creatures. Possibly either there's a dog model and also there is sheep and a couple of other farm animals that can show muscular dystrophy from time to time. So in their muscles, what's happening is there are very few mitochondria. See these thick mitochondria, they form kind of a continuum. We don't see them hardly at all here by the sarcolemma. And that means someone with muscular dystrophy, and if we look at someone who's older, we almost see the same thing. We don't see as many mitochondria, especially near the cell membrane. They've been damaged or removed usually because they've been damaged. That means patients with muscular dystrophy, someone who's older, is going to have, because there aren't enough mitochondria, fairly poor endurance. So even if they aren't strong, they're also not very fatigue resistant. So that becomes a big problem. So we need intact dystrophin and nNOS, it appears, for these mitochondria in order to work properly. So when muscles produce tension or force, there are two ways they do that. One is by something we call passive tension. And by passive tension, I mean that if we stretch a muscle, especially if we do it rapidly, it stores energy and then it recoils. Boom. So when a muscle is pulled, when it's stretched rapidly, it's going to try to produce force. That's a recoil force. So like stretching a rubber band, or if you have a slingshot, pulling it back, or a bow and arrow, it's the same kind of thing. So the muscle's going to try to respond. So this is measuring, and this is at like a molecule level. They're looking at a protein called titin, which is sometimes called the spring protein. And they're looking here at the heart and skeletal muscle. Titin is determining a lot of the passive tension inside the muscle fiber. The other type of force production is active. And so that means excitation, contraction, coupling. And action potential comes down the sarcolemma. So it was originally in the alpha motor neurons at the neuromuscular junction. Acetylcholine is released that binds to its receptors. That causes sodium to rush in and depolarize the cell. The action potential is carried down into the T tubules, where there's coupling between a voltage sensing calcium channel called a dihydropyridine receptor, and then a sarcoplasmic reticulum calcium channel called a ryanodine receptor. In skeletal muscle, these are mechanically linked. In the heart, you actually need a calcium spark in order to release calcium from the sarcoplasmic reticulum. So calcium is involved in what we would call the feed forward or a positive feedback cycle. I would say that's a feed forward system. So, but in skeletal muscles and the heart, you release calcium. Calcium binds to a protein called troponin. Troponin causes tropomyosin, that's TM, to roll over and release a binding site so myosin can activate it. So this is called excitation, contraction, coupling. We're going to talk in a few minutes about the sliding filament theory. And that allows us to be able to explain why when muscles shorten, you have these cross bridges being formed again and again and again. And as it shortens more and more, we get to the point where the actin and myosin filaments will overlap a little bit, and we won't get optimal number of binding sites. It's really all about how many binding sites between actin and myosin you can form. The more binding sites you can produce, the greater the amount of force. The faster we can cross-bridge cycle, the faster the muscle can shorten or contract. So passive tension, we think, is like tendons and ligaments and bones, a property of the materials in them. So it's really titin and other proteins inside the muscle fibers and the connective tissue. That combination of the muscle fiber and connective tissue produces passive tension. Again, working really just like a rubber band. If we look at, and this is a load elongation curve, which is a lot like a stress strain curve, we can see that as you stretch a muscle faster, it gets stiffer faster and can produce more force. So when a muscle is stretched fast, it is elastic. It's going to basically store a great deal of force. So that happens when we're doing a wind-up, for example. We want things to be elastic. But if we stretch a muscle slowly, and we can take advantage of this when you're doing like stretching exercises. You want to do those slowly. You don't want to do ballistic stretching, because ballistic stretching, first of all, will activate the muscle spindle reflex, which of course will produce more active tension in the muscle you're trying to stretch. Secondly, the muscle produces more passive tension when you stretch it fast. When you stretch it slowly, it becomes viscous. Viscous is the opposite of elastic for a fluid, and it means we're, instead of storing energy, like elastic materials do, we are going to dissipate energy. We're going to get rid of energy. That may not sound like a good thing, but it is when you're stretching. You want that muscle to flow, almost like molasses. So that's an example of a material that would be viscous. Okay, so that's active and passive tension. When we put the two together, we can simply add them together. So curve A is a passive tension curve. Curve B is actually the combination of the two. The dotted line is active tension. So if we add together passive tension plus active tension, we're going to get total tension. And it's going to peak at about 120% of resting length, meaning just a little past the normal resting length of a muscle. If I just dangle my arm like that, my biceps are probably at resting length. If I go close to full extension, my biceps brachii are probably pretty close to 120% of resting length. In other words, an extra 20%. And when we see that, we see this bump in peak tension. That's going to be near the end of the range of motion, right in here is where that occurs physiologically. We can get muscles to stretch further, but we wouldn't normally see this physiologically inside a person. Okay, so again, a reminder that passive tension is caused by the material properties of the muscles and the connective tissue. Titin is the spring protein, so we think it's one of the major activators of passive tension. It didn't really sound right, did it? An activator of passive tension. The major component of passive tension. The connective tissue as well in the endomysium is the other really important component. The farther we stretch the muscle, the greater the amount of passive tension. The faster we stretch the muscle, the greater the amount of passive tension. It's also true that there's greater amount of muscle tension as well. So here's the active tension curve. Here's the passive tension curve. If you put the two together, the peak is right about here. And again, that's very close to the end of the range of motion. So at this point, we want our biceps to be able to produce lots of force. What they're trying to do is hold that elbow together and keep it really stable. So near the end of the range of motion, our body's not as interested in producing torque. It's more interested in providing protection. So that's exactly what we see. Okay, so when a muscle is contracting, and well, let me back up the truck. When a muscle is producing passive tension, it's doing so through, again, a protein called titin. And titin covers from, and this is an entire sarcomere. We have the Z disc on either end, then we have the M line in the middle. The protein titin attaches the M line to the Z disc, and it's got kind of a spring built in. There's literally part of the protein that can act like a spring. It actually has a name that we won't worry about because so when a muscle is being stretched, this spring is being stretched, much like a muscle. In fact, it's why titin's sometimes called the spring protein. When the muscle is being stretched or a rubber band is being stretched, then it's going to have a tendency to recoil, especially if in a muscle the stretch is fairly rapid. So here's titin. Notice it's right next to the actin filament. And then both of them bind to the Z disc. So this is what we would call the thin filament, made of actin. The thick filaments are made of myosin. So in this excitation contraction coupling, what you have is binding of myosin heads to binding sites on actin, and that produces the cross bridge. Okay, so again, with passive tension, when we look at a tension length curve, and you have tension on the y-axis, length of the muscle will be on the x-axis. So we're stretching the muscle as we go from left to right, and as we stretch it, we see passive tension increasing. In fact, if we looked at the material properties of a rubber band, they look almost exactly like that. At first, they're going to have some slack built in, and then as you stretch them, they get stiffer and stiffer and stiffer. And you probably get to the point where they don't really want to stretch anymore. If you snap them, you feel that energy being released. You can actually store a significant amount of energy in a muscle or in a rubber band. As we stretch the muscle, we get, again, close to the end of the range of motion. We're building this passive tension. We're also getting an increase in active tension. The product of the two, or I should say the addition of the two, the sum of the two, gives you total tension, the sum of those two. These are real muscles from real animals, real people, I think. 29-year-old, these are real people. So they've been able to look at stress strain curves at real human muscles. And again, these curves remind me a lot of a rubber band. As we start to stretch them past 20, 30%, this is getting close to the end of the range of motion right here. If you were wondering, by the way, right in this area is probably where we're going to be. So you notice as they muscle stretch, you get an increase in the amount of tension. And each muscle is a little different. Some have more connective tissue than others. The sartorius is a thin muscle, so it actually has more connective tissue. Your biceps brachii has most of the fibers in parallel, and they're surrounded by significant connective tissue. So is the rectus femoris. That means it gets stiffer a little earlier on this curve. The gastrocnemius is a pennate muscle. The pectoralis major is kind of fan-shaped, and both of those lead to a situation where connective tissue is not contributing quite as much, so they're not quite as stiff, and that's why they're on the right side of the curve. But regardless of which muscle it is, notice as you increase muscle length, you get this increase in slope. Again, that happens just like what we see with a rubber band. Okay, so to kind of sum up passive tension and recoil, when a muscle is stretched, think of it as like a happy muscle, it's able to store a significant amount of force and energy. As it recoils, you can take advantage of this with a windup. When a muscle is stretched slowly, it's viscous. It dissipates force and energy. So stretching that muscle to increase flexibility, range of motion is part of a warm-up exercise, especially as part of a cool-down exercise. Muscles are actually, it's more effective to stretch them after exercise, believe it or not, than before, because the muscles are more compliant, they stretch more easily. You can get better flexibility gains afterwards, or even in the middle of a practice. When a muscle is stretched slowly and it becomes viscous, it can produce something called hysteresis, which sounds frightening a little bit. But what hysteresis is, it just means that when a muscle is being loaded, and then when it unloads, the shape of that curve between force and length is not going to be the same. It's going to relax more on the way back. And that means it's becoming viscous. Instead of storing energy and recoiling, again, like the rubber band example, watch out, it's going to relax, much like your lungs do when you breathe in. When you breathe in, it's elastic. When you breathe out, then your lungs become more viscous, and that allows the bronchioles to remain open. If your lungs were extremely elastic breathing on the way out, especially if you're breathing relatively slowly, then what would happen is they would squeeze on those bronchioles and reduce your airflow out. And reducing airflow out is actually what happens when we have people who have asthma. So it's breathing out that's more of a problem than breathing in. Okay, hang on there. A little technical problem. Let's see if we can fix this. Oh, there we go. Okay, good. We're back. So the speed of a muscle's contraction or speed of, I'm probably going to add to this, stretch, does make a big difference. When stretch muscles, again, are rapid, it's elastic. If it's slow, it's what we would call viscous. The area in between the loading and unloading curves, that's called hysteresis, what we were just talking about. This is the energy that's actually released or not absorbed. Therefore, at the end of the story is from passive tension. When muscle length increases, we increase the amount of passive tension. It can add to active tension that's being formed through excitation, contraction, coupling, and that yields total tension on a muscle. And where we see this kind of bump at about 120% of resting length, that's where active tension and passive tension are combining to really max out near that end of the range of motion. So for the biceps brachii, when the elbow joint is close to full extension, that's where we actually see the greatest amount of force development in that muscle. And that could protect, let's say you're lifting a heavy bag of groceries, or you're lifting a suitcase or a heavy object, and your elbow's extended. That biceps brachii is not doing so well in terms of producing torque, but it's really effective in terms of maximizing the support and the pressure being produced at the joint between the articulating bones, and therefore maximizing the amount of stabilization. So when muscles are stretched, you can have passive elastic recoil. If the muscles stretch rapidly, if it's stretched slowly, then not so much. Okay, so this is the end of the material for test number two. We are going to take a break for a moment and then start talking about active tension. We'll start talking about active tension. I think I may split this particular lesson into two parts, because once I stop, it's just easier to save it, and then I'll come back and start the next session. Okay, howdy, we are back. We're going to start material related to active tension. So this will, starting right here, this material will be on the final exam. So the final exam will include the last third of the class, then another 50 points will be cumulative, but some of it could be from this third of the class. Skeletal muscle mechanics are an important part of not only the class, but being a kinesiology major and the material you need to use to be able to understand how musculoskeletal systems work. And that's, I think, critically important for a kinesiology major. To me, that's what it's about. How do musculoskeletal systems work? How does your cardiovascular system work during exercise? That, to me, covers a lot of what's going on. And that's going to include things like metabolism that'll help to be able to explain why exercise, for example, can reduce the risk of type 2 diabetes, obesity, etc. So I hope you're getting your exercise. In fact, in a few minutes, I'm going to take a break and try to go get some exercise myself, and then come back and finish the second part of today's lecture. That'll be pretty short. Okay, so we're going to talk about excitation, contraction, coupling, and just review that in the next few minutes. We'll take another little break. We're then going to cover muscle size and muscle length, maybe ray coding, and then the rest of the lecture will on tomorrow, the second the second lecture of the week, we'll cover some of the rest of these mechanisms that lead towards an increase in active tension. And we'll then kind of sum things up with skeletal muscle by looking at the effects of aging, injury, overuse, even delayed onset muscle soreness, etc. Okay, so excitation, contraction, coupling. This is how we turn on a muscle. It's the engine that provides force and power for the body to be able to propel itself forward. So it's like a car or an automobile or a truck engine. So excitation, contraction, coupling, that notion came about in the 1950s. And so we're going to look at some of the scientists very briefly who were involved in some of that research. This is a reminder that muscles and the alpha motor neurons work together as what we call a motor unit. Each alpha motor neuron, and that should be there, should be that. There we go. Boom. That's my little symbol. Alpha motor neuron can innervate anywhere between 20 to 2,000 fibers. For muscles that operate eye movement or finger movement, we may have 20, 40, 50, 100 fibers per motor unit. This gives us fine motor control. So by having fewer fibers per motor unit, it means you get more switches. You can control things like muscle movement. In a room, you can control light more easily. The more switches you have, the better fine-tuned control you have. Large muscles like the quadriceps, the gastrocnemius, and the gluteus maximus may have 1,000 fibers or more per motor unit. A, they have a lot more fibers, and B, we're interested in gross motor control rather than fine motor control. So each motor unit is made of an alpha motor neuron and all the fibers that it innervates. So this motor unit number 2, innervates this fiber, innervates the second fiber from the left, or from the, I guess, second fiber from the left. So each of these fibers actually would be the same relatively in terms of fiber type, either fast twitch or slow twitch. Their histology would be roughly the same. So would their biochemistry, their ability to resist fatigue, the relative size or cross-sectional area of the muscle fiber itself. So different motor units will innervate different fibers, again, anywhere between 20 and 200. And again, they control the fiber type, they control the biochemistry, they control the histology of the cell itself, how it's made up. There was a famous study done at the University of North Carolina many years ago, probably about 70 years ago, where they switched the motor units. Let's say you had a fast twitch motor unit and then the slow twitch motor unit. So the slow twitch alpha motor neuron innervated slow twitch fibers. The fast twitch alpha motor neuron innervated fast twitch fibers. If you switch the motor neurons, then what happened was the muscle fibers migrated from slow twitch to fast twitch and fast twitch to slow twitch. So that's how we knew that the alpha motor neuron controls a lot of the makeup and histology and biochemistry of those muscle fibers. When action potentials are triggered down these alpha motor neurons, they go to the neuromuscular junction where their quanta of acetylcholine that are released. Acetylcholine binds to the receptors on the cell membrane. That allows sodium to rush in, and that initiates this process we know as excitation, contraction, coupling. So you have action potentials that travel down the alpha motor neuron, release the neurotransmitter acetylcholine, fixing things as we move through. The action potentials conducted down the sarcolemma and then into the T tubules, where they can trigger a voltage-mediated calcium channel called the dihydropyridine receptor. It's going to pass that information on to the sarcoplasmic reticulum, where we have specific calcium channels that release calcium that's stored in the sarcoplasmic reticulum. That increases the concentration of calcium in the cell. It goes from nanomolar level to millimolar level. So we're looking at about 5 orders of magnitude difference, huge, like 10,000-fold increase. Then the calcium binds to troponin C, and that causes a change in shape of troponin. Tropomyosin is lifted, or really what happens is it rolls over, and it reveals the binding site between actin and myosin, and that's where cross bridges form. The myosin heads have been activated when they were released off of the last binding site. So they're basically stored with energy. They have potential energy built in. When tropomyosin rolls off, those myosin molecules can now bind to actin. And when they bind to actin, they're going to start to pull. They have this potential energy. As soon as they hit the binding site, they start to pull. So cross-bridge cycling means that these myosin heads, and there are millions of them in a muscle cell, they'll release, and when they release, that takes ATP. That's where the energy is being used. When they release, and then they're going to go upstream, pull again, go upstream, pull again. This is called cross-bridge cycling. So think of someone paddling, like on the water. Let's see. Sorry about that. My paddle's migrated. So if someone is paddling on the water, you have to pretend this tennis racket's a paddle. That's what cross-bridge cycling is like. So you bind to the actin site, pull. That's where you actually use the ATP, lift off. You have that potential energy, and then pull. Keep pulling. The faster you can cycle, the faster a muscle can shorten. However, it does mean there's a sacrifice in terms of force development. The slower you're moving, like when you're doing a max bench press, you probably are not lifting it very fast, but that's the trade-off. You can produce now a lot of force. If you're moving really fast, you can't produce nearly as much force. That's because a lot of the cross bridges are in the middle of the cycle. The myosin heads are lifted off of the binding sites. When you have an isometric contraction, that means you're maximizing the number of myosin molecules or myosin heads on those binding sites. The more cross bridges you have at any moment, the more force you can produce. But to get shortening, you got to move them. So that means with that turnover, that's a trade-off between force development and speed. So the myosin heads combined with the actin molecule, they pull, and the cross-bridge cycling continues in the presence of calcium and ATP. One of the reasons we know ATP is needed to detach the myosin heads from actin is when rigor mortis sets in. So with rigor mortis, what happens is there's calcium that leaks into the cell. Tropomyosin rolls off. The binding site's there. The myosin heads attach. They are pulling, but there's no ATP to release them because metabolic enzymes, those type of proteins, are the most labile, and they'll go first after a person passes away. So 7 or 8 hours later, you got rigor mortis because all of those myosin heads are bound to those actin molecules. Let's click on the right thing. That's not it. Okay. Let's go down here. There we go. So again, with excitation, contraction, coupling, we have depolarization of the cell membrane. And that happens within just 2 milliseconds. It's really fast. Then we're going to see some of that signal or depolarization go down into the muscle fiber through the T tubules, where the dihydropyridine receptor is, and that's going to directly connect in skeletal muscle to the ryanodine receptor in the sarcoplasmic reticulum. So there we go. This is a reminder. Muscle fibers are made up of a series of sarcomeres. The cross bridges formed are between actin and myosin. You actually have myosin heads. And this notion, or sliding filament theory, came from a couple of Huxleys. I don't think they're related, if I remember correctly. Andrew Huxley of Cambridge worked with Alan Hodgkin to first come up with the theory of excitation, contraction, coupling in the early 1950s. Hugh Huxley was involved in the sliding filament theory and the cross-bridge mechanism later in the 1960s. So the two Huxleys, along with Hodgkin, they were involved in this particular model. And it turns out, even at the molecular level, we've seen that this model is correct. So it means that there are myosin heads that can bind to binding sites here with actin. When a muscle is shortened, though, some of that, there's some overlap, and these binding sites, or the myosin heads, are no longer near the ideal or optimal binding sites on actin. So it means you have fewer cross bridges. With fewer cross bridges, you have less force production. So with less force production, we did this little exercise where we flexed our wrists like that and then try to grab a pencil or pen. We kind of noticed, oh, that was really difficult. So with active tension, as we shorten the muscle, we reduce the number of cross bridges that are available. So with muscle contractions, it means we're trying to shorten the muscle. If the muscle can produce more torque, then the torque is trying to overcome due to a weight, due to antagonist muscles, due to impact forces, etc. So something really interesting when a muscle contracts, like when people make a muscle, right? When they do that, notice that muscle, oh boy, it looks like it's getting big. So as the muscle gets shorter, the water has to go somewhere because water is an incompressible fluid. Unlike air, you can compress air like you can in a piston. Air is what we call adiabatic. So this is a hydraulic effect. And it means that the water goes sideways and actually provides support for the connective tissue and the muscle fibers. That's why it's actually hard to produce a lot of muscle damage with just a concentric contraction. You need eccentric contractions to produce damage, because then water doesn't actually stabilize the muscle fibers nearly as well. So what I'm saying is the water is pressuring sideways, pressing on the connective tissue together, sandwiching it so it doesn't produce too much shear force. So it's actually a protective mechanism. When a muscle is being stretched, though, you don't have that protective mechanism. That muscle is just lengthening. The water is, instead of pushing on the fibers, it's sliding across the fibers. And you sometimes have hot spots for too much shear stress, which can lead to damage. So as we were saying, in excitation, contraction, coupling, in skeletal muscles, where we pass kind of the mechanical ball, is when there's depolarization of the T tubules. The dihydropyridine receptor, this is a voltage sensor, if you will. It's a special type of calcium channel. It directly binds to the ryanodine receptor, and there's a few other triad proteins in there, and that's going to release calcium. And as calcium concentration increases, that's going to increase the force development of a muscle as the cross-bridge number increases. So it's a direct linkage. In the heart, let me add this, there we go. In the heart, you need actually small calcium currents called calcium sparks to communicate between the dihydropyridine receptor and the ryanodine receptor. So there's literally a gap in heart muscle, but there's not in skeletal muscle. So skeletal muscle, the connection is mechanical. In the heart, it actually is through what we call calcium-induced calcium release. And that just means the calcium that shoots out of this voltage sensor stimulates the ryanodine receptor. In the case of skeletal muscles, it's actually the protein itself is getting this change in shape, which is triggering the ryanodine receptor to release that calcium. So when cross bridges are formed, tropomyosin rolls off the binding site. We have a pair of myosin heads, and they're going to start pulling right away. They have potential energy built in. And as long as they continue to pull, you get a continued cross-bridge cycling. The faster the cycling, the greater the velocity, but there's less force they can produce. So that, again, is the trade-off. So we're going to pick up right here with cross-bridge cycling. We're going to say there's enough calcium. Calcium's caused troponin and tropomyosin to reveal the binding site. The myosin heads are right there. The cross bridge forms, and the myosin head's going to pull right away. So that's one of the reasons we know muscles produce tension, besides the fact that tendons can't transfer tension anyway very well. The reason we know at the molecular level is these myosin heads, when they hit that binding site, they're going to start pulling right away, just like a kayaker. So that's where the action is. This is what we call the power stroke. Then we've got to release, and that's going to take ATP to do it, and we're going to start again. And each time your muscle is trying to shorten, so you're going to move many binding sites, attach again, and pull. There are hundreds of thousands of these myosin heads binding at any point in time when your muscle's contracting. And as it's shortening, they've got to cycle really fast. The faster they're cycling, the greater the speed, but the less force your muscle can produce. The less force your muscle can produce. Okay. So we just said it's all about the number of binding sites between actin and myosin. We're going to see that demonstrated when we talk about muscle size or cross-sectional area, muscle length, stretching a muscle. So both of those will increase the number of cross bridges and increase the amount of force that a muscle can produce. Okay, so we're going to take another break. I'm going to do some running, etc. Come back, and then we're going to finish this part of the lecture. It'll probably be in two parts as you guys are looking at it on eCampus. All right, so we'll see you in just a few minutes.